হ্যাঁ,
ম্যাম আমি না ম্যাম 7 বেসেরও আছে বলতেছে।
ম্যাম
7
71।
আচ্ছা ঠিক আছে, অসুবিধা নাই। 7 ব্যাচে যারা আছো এটা আলাদা লিস্ট, এখানে শুধু 8 ব্যাচ আমি কাউন্ট করছি, ঠিক আছে? আচ্ছা, ওকে। We are continuing about Viva question, discussion of Viva question, digestive physiology. In previous class, we discussed, we have started about pancreatic juice. Again, I want to discuss from here. Pancreatic juice, composition of human pancreatic juice for written exam and for viva exam. For written exam, you have to write everything, that is water 98.5%, solid 1.5%, inorganic constituents, that is cation and anions, and organic constituents means digestive enzymes. But for viva exam, what are the important constituents for pancreatic juice? You have to mention only from here: proteolytic enzyme, carbohydrate splitting enzyme, and fat splitting enzyme. These three things very important for viva exam. But written exam, you have to write everything. For viva exam, proteolytic enzyme, name of the proteolytic enzymes of pancreatic juice. As proenzymes: trypsinogen, chymotrypsinogen, procarboxypeptidase A and B, proelastase. These are the proenzymes. But active enzyme, ribonuclease and deoxyribonuclease, these are the active enzymes. Okay. Now, carbohydrate splitting enzyme or amylolytic enzyme, pancreatic alpha amylase, fat splitting enzyme or lipolytic enzymes, pancreatic lipase, cholesterol ester hydrolases, phospholipase A2, inactive form, and procolipase. This is also inactive form, that is proenzyme form. Okay. These are the important constituents of pancreatic juice. Okay. Now, factors that prevent autodigestion of pancreas. This is very, very important viva question. Why the autodigestion of pancreas is not occur? Due to what reason? See here, the proteolytic enzymes of the pancreatic juice are secreted always as inactive form. That is as zymogens. That is trypsinogen, chymotrypsinogen, procarboxypeptidase, and proelastase, that is proteolytic enzyme of the pancreatic juice, are secreted as inactive form, that is zymogen. So they are not activated until they are secreted into the intestine. That is, they are activated when they come into the intestine, that is duodenum, upper part of the small intestine. That is when they come into the duodenum. In duodenum, there is one enzyme present, that is enterokinase, which causes activation of this inactivated enzyme is activated by the presence of enterokinase. This enterokinase enzyme present in the upper small intestine, that is in the duodenum. So the proteolytic enzyme always secreted in the pancreatic juice are inactive form. This is one reason. So inactive trypsinogen and other enzymes cannot digest the pancreas. If these enzymes secreted as active form, they digest the pancreas. That is, autodigestion of pancreas will happen that time, but they always secreted as inactive form. Now, along with the enzymes, pancreas can secrete a one inhibitor, that is trypsin inhibitor, that always prevent the activation of trypsin. That is, inactivated pancreas or pancreatic juice secrete inactivated enzymes, that is proteolytic enzymes, trypsinogen, chymotrypsinogen as inactive form. Also, the secret trypsin inhibitor. That is, this inhibitor always prevent activation of trypsin. These two reasons why autodigestion of pancreas not occur, due to two reasons. One thing, proteolytic enzyme always secreted in the pancreatic juice in inactive form. This is one reason. And another reason, that is pancreas also secrete one thing, that is trypsin inhibitor. It always prevents the activation of trypsin. So these two reasons prevent autodigestion of pancreas. Okay, this is very popular viva question. Why autodigestion of pancreas is not occur? Now, How activation occur? Activation of pancreatic proenzymes, already I mentioned, the pancreatic proenzymes are activated in the intestinal lumen, that is intestinal mucosa secretes enteropeptidase or enterokinase, that is in the upper small intestine, that is in the duodenal mucosa secretes enterokinase or enteropeptidase, two things, same thing, enterokinase or enteropeptidase, same thing. These activate the proenzymes, that is pancreatic proenzymes. See here, proenzymes and enzymes. Trypsinogen, this is proenzymes or zymogen. Trypsinogen is activated by enterokinase in the upper small intestine, that is in the duodenum, and converted to trypsin. Whenever trypsin converted, this now trypsinogen, this trypsinogen, by the presence of trypsin, it converted to trypsin. Again, chymotrypsinogen, in presence of trypsin, it converted to chymotrypsin. And pro-elastase, in presence of trypsin, it is converted to elastase. Pro-carboxypeptidase, in presence of trypsin, it is converted to carboxypeptidase. That is, first reaction is very, very important. Trypsinogen, in presence of enterokinase, it should be converted to trypsin, otherwise all reaction will stop. Okay, this is the real thing. Now, types of pancreatic secretion, already we discussed this thing. Again, I want to recapitulate this thing because this slide is very important for written MCQ and Viva. Okay, hydrolytic type and ecbolic type. What do you mean by hydrolytic type? H for H, that is pancreatic juice secretion rich in bicarbonate, but water, rich in bicarbonate and water, but poor in enzymes by the action of secretin, because of action of secretin. This is very important thing. Secretin, always secretion is water is secretion. Here copious secretion occurs here, and this juice is rich in bicarbonate. The bicarbonate and water, that is copious water is secretion. That is secretin, hormone secretin is responsible for copious watery secretion and rich in bicarbonate secretion of pancreatic juice, that is hydrolytic type of secretion. Okay, now ecbolic type, E for E, pancreatic juice secretion rich in enzymes but poor in bicarbonate by the action of cholecystokinin. Cholecystokinin, it has the digestive function. That is pancreatic juice when rich in enzymes but poor in bicarbonate, that is ecbolic type. Now, what is the differences between hydrolytic and ecbolic type of secretion? Hydrolytic type, it is rich in bicarbonate but poor in enzymes. Already you know this thing. Ecbolic type, it is rich in enzymes but poor in bicarbonate. And hydrolytic type, it is thin and watery, it is thick. Okay, its secretion is stimulated by secretin. Always remember this thing. Secretin is secretion. For copious secretion, water is secretion, and bicarbonate rich secretion. Okay, and it is this secretion is stimulated by cholecystokinin and acetylcholine. Very, very important MCQ question. Hydrolytic type secretion is stimulated by secretin, and ecbolic type secretion is stimulated by cholecystokinin and acetylcholine. Now, its secretion in response to acid chyme in the duodenum. Acid chyme in the duodenum. This is very important MCQ question. Hydrolytic type secretion is response to acid chyme in the duodenum. In response to acid chyme in the duodenum, and secretion occurs in response to fatty food in the duodenum. It mainly neutralizes the acidity of the chyme. It is digestive in function. These are the differences between hydrolytic type and ecbolic type of secretion. Now, regulation of pancreatic juice secretion, very, very important SCQ question, written question. Several times this question came under Chittagong University and Chittagong Medical University. Several times this question came. So you have to write down in your khata like this way. Regulation of pancreatic juice secretion. See here, there are two types of regulation: nervous regulation via vagus nerve and hormonal regulation by secretin and cholecystokinin. Nervous regulation via vagus nerve and hormonal regulation by secretin and cholecystokinin. See here, phases of pancreatic secretion. There are three phases included in pancreatic secretion, that is cephalic phase, gastric phase, and intestinal phase, like gastric juice secretion, like gastric juice, cephalic phase, gastric phase, and intestinal phase. Both two phases, cephalic phase and gastric phase, always occurs via vagus nerve, but intestinal phase occurs by secretin and cholecystokinin. There are both first two phase, cephalic phase and gastric phase, occurs by nervous regulation, that is via vagus nerve, and intestinal phase occurs mainly by hormonal regulation, that is secretin and cholecystokinin. Three basic stimuli are important in causing pancreatic secretion, that is acetylcholine acts on acinar cells of pancreas, cholecystokinin acts on acinar cells of pancreas, secretin acts on ductal cell of pancreas. This slide is very important for MCQ exam. Acetylcholine acts as acinar cells of pancreas, cholecystokinin acts as acinar cells of pancreas, secretin acts on ductal cells of pancreas. Now, hormonal mechanism, mainly intestinal phase, already mentioned, intestinal phase mainly dependent on hormonal mechanism, that is secretin and cholecystokinin. That is, it is the most important mechanism for secretion of pancreatic juice. It occurs in intestinal phase by two local hormone, that is secretin and cholecystokinin. How this mechanism occurs: when acidic chyme comes into the duodenum, release of secretin by the duodenal mucosa, secretin goes to pancreas via blood, acts on the pancreatic duct, release of pancreatic juice rich in bicarbonate but poor in enzyme. That is copious watery secretion but rich in bicarbonate, but poor in enzymes. This is hydrolytic type of secretion, which is hormone here responsible, of course, secretin. Hormone here responsible, of course, secretin. Now, when fat and protein comes into the duodenum, release of cholecystokinin by duodenal mucosa, this cholecystokinin goes to the pancreas via blood, acts on the pancreatic acini, release of pancreatic juice rich in enzyme but poor in bicarbonate, that is ecbolic type of secretion. These are the mechanisms of hormonal mechanisms. These are the hormonal mechanism in intestinal phase. Okay, you have to draw this figure in your written exam, regulation of pancreatic secretion. From your textbook guidance 64-10, see here, acid from stomach releases secretin from wall of the duodenum. Fats and amino acid causes release of cholecystokinin. You know that very well, that secretin always secrete when acid comes from stomach into the duodenum, and fatty food when comes into the duodenum, that releases cholecystokinin. Okay, vagal stimulation releases enzymes into the acini, and secretin causes copious, of course, secretin always causes copious secretion of pancreatic fluid, that is watery fluid rich in bicarbonate, and cholecystokinin causes secretion of enzymes, that is enzyme rich fluid. And enzyme rich pancreatic juice secretion occurs by cholecystokinin enzyme. Now, secretin and cholecystokinin absorbed into bloodstream. These are the figure, regulation of pancreatic secretion. You have to draw this figure in your written khata. Okay, now this is short note. Several times this question came in your exam, first prof exam. What is, write short note about steatorrhea? Steatorrhea. What do you mean by steatorrhea and causes of steatorrhea? Steatorrhea means pancreatomized animals. Pancreatomized animals, that is pancreatectomy occurs in those animals. Pancreatectomy means whenever you cut pancreas. Whenever you cut pancreas, this is called pancreatectomy. In case of pancreatomized animals and patients with disease that destroy the exocrine portion of the pancreas, have fatty, bulky, clay-colored, foul-smelling, greasy stool is known as steatorrhea. যদি কোনো অ্যানিমেল অথবা কোনো পেশেন্টের দেখা যায় যে প্যানক্রিয়াসটা যদি আমরা কেটে ফেলি, ঠিক আছে, প্যানক্রিয়াটমি যদি করে ফেলা হয়, তাহলে প্যানক্রিয়াটমি মিন্স কি, কাট ডাউন, তাহলে প্যানক্রিয়াস যদি না থাকে অথবা দ্য ডিজিজ দ্যাট ডেস্ট্রয় দ্য এক্সোক্রাইন পোর্শন অফ দ্য প্যানক্রিয়াস, তাহলে কি হবে, ফেটি, বাল্কি, ক্লে মানে কি, হোয়াইট কালার, সাদা, ক্লে কালার্ড, ফাউল স্মেলিং, একটা গন্ধযুক্ত, এবং গ্রিজি স্টুল মানে কি, একটু দেখা যায় যে ফেটি স্টুলটা একটু গ্রিজি স্টুল ইজ নোন অ্যাজ স্টিয়াটোরিয়া। Causes of steatorrhea key. First one, malabsorption syndrome. Number 2, lipase deficiency. Number 3, hypersecretion of gastric acid. Number 4, defective reabsorption of bile salt in the distal ileum. These are the causes of steatorrhea. Okay, these are the causes of steatorrhea. Now, composition of Succus entericus. Composition of Succus entericus: water 98.5%, solid 1.5%, inorganic, that is cation and anion, sodium, potassium, calcium, magnesium, these are the cation, anion, chloride, bicarbonate, phosphate. Organic enzymes. This is very important for your Viva exam. Important constituents of Succus entericus, you have to start from here, that is from proteolytic enzymes for Viva exam. For written exam, you have to write down everything from water, solid, everything. But for Viva exam, important constituents, proteolytic enzyme, you have to start from here. That is aminopeptidase, dipeptidase, tripeptidase. Amylolytic or carbohydrate splitting enzyme, maltase, sucrase, isomaltase, lactase. Lipolytic enzyme, intestinal lipase. Activator enzyme is enterokinase, of course, enteropeptidase, and also another constituent, mucus. Then the first thing, organic components, enzymes, and number 2 is mucus. These are the important constituents of Succus entericus. Succus entericus, or small intestinal secretion. Okay. Now, what is cholegogues and choleretics? I want to recapitulate again. What is cholegogues? Substances that causes contraction of the gallbladder are called cholegogues. They increase the flow of bile. All cholegogues are choleretics. That is cholecystokinin, gastrin, acetylcholine. These are the cholegogues. Cholegogues কি করে? Contraction করে. Choleretics কি substances that enhance excretion are called choleretics. They increases the production of bile. All choleretics are not cholegogues. This is MCQ question. Very important MCQ question. All choleretics are not cholegogues. Bile salt and bile acids are the major physiologic choleretics. Others are stimulation of the vagus nerve, hormone secretin, and cholecystokinin. Okay. Okay, that's all from me today. Thank you, everybody. Thank you very much. Next class we'll discuss about bile. আমরা biliary secretion নিয়ে কথা বলবো, gallbladder bile, liver bile, enterohepatic circulation, ঠিক আছে, functions of liver function test we'll discuss in the next class. Okay, thank you everybody, thank you very much.